Good morning. I spent my whole morning working through poster drafts and it was fantastic. Many of them are so good, and I am sending back what I hope is substantive feedback to enable you to make some really good changes before hitting print. Remember that I won't have time to assess your posters for a second time, so once you've incorporated all of my suggestions, just know that that will improve your scores in each rubric category, and you can be ready to present them on Monday of finals week. So remember that on Monday of finals week, we will be presenting our posters in the foyer of the NZ STEM building. You're going to want to arrive just a little bit early, or at least one member of your group. Try to get there maybe around 9:30 so that you can set up and be ready for that 10:15 start time. We will be taping our posters to our usual favorite writing boards, and hoping that everyone will come and circulate around the room and talk with us about our posters. Half of you will present, quote unquote, between 10:15 and 11:15, and half of you will present in the latter time, 11:15 to 12:15. And that information as to which time you present will be found on the abstract packet, which I will be working on, and I will be hoping to get you a draft of the abstract packet by Thursday night or Friday morning so that we can get that all sent around to faculty and invite them and be ready to present to a broad audience come Monday. So that being said, I think that's most of the information that you need to know, except for that you will need to turn your final exam in sometime prior to or on Tuesday morning. And I'm looking towards either your individual submissions or your group submissions for that final. Please remember that if you choose the individual submission route, your words need to be your own. So you need to make sure that even though you're getting help from all your peers and everyone else, that when you sit down to do the writing, that you write on your own and take it home alone with you, and make sure that it's your words that I hear and not someone else's. Okay, I think that takes care of nearly everything that we need to talk about logistic-wise, but certainly we have a lot more to talk about content-wise and lots of exciting stuff. We're going to be ending the day with a tour through Yellowstone, but first we need to remind ourselves of where we left off last time. We were looking at both phylogenetic and phonetic methods of classification, and we had talked about how you can utilize DNA hybridization to determine the species level, but we also then called our attention to the fact that that's not always even the perfect method itself. So it's probably true that using multiple methods is the way to go. And I want to end our coverage of taxonomy by looking at the use of 16S ribosomal RNAs for the purpose of classification. We come back to this again and again because really this is the gold standard at this point for building phylogenetic trees and looking at comparisons between these sequences, because they can serve as molecular chronometers. That means that over time they tick off the changes and they can show us divergence from a common ancestor. And really this is what gave rise to Carl Woe's phylogenetic tree that we spent some time talking about, and what has continued to lead to the evolution of a more descriptive phylogenetic tree. And in fact, since we have now been able to get whole genome sequencing for many microorganisms, the use of whole genome sequencing and metagenomics and the rich data that we're getting from that has really been what's led to the synthesis of this phylogenetic tree that was published in Nature not so long ago, looking at the very large increase in the number of bacterial species that we can identify based upon all of the new genotypic methods that we have accessible to us. And I did call your attention to this tree a while back, but what maybe I didn't fully call your attention to is the incredible over-representation of life that sits within the bacterial domain of life. Maybe over-representation is the wrong word. It's an acknowledgement that bacteria are the dominant life form on Earth. Remember reading through Stephen Jay Gould's passage about the fact that bacteria are in fact the dominant form of life. Well, this wonderful figure really allows us to see that, because notice that this is the bacterial domain here. And then if we look down here, this is archaea right here, and then here we are eukaryotes. So we get that sense that yes, bacteria are most certainly the dominant form of life on Earth. And so this is a beautiful segue to the rest of our coverage, not only of phylogenetic trees, which that's a beautiful one, but also to our consideration of Bergeys Manual of Systematic Bacteriology, which in a lot of ways, it's like the epitome source for looking at what we know exists. And of course, it's becoming outdated every single day. So we know that there's something added all the time that's not going to be even in the most recent edition of Bergeys Manual. But it is a set of books that allows us to look up information on those that are known and learn more about them. So let's take a look first at examples of phylogenetic trees and whether or not we're looking at ancestry or we're just looking at relatedness. So rooted trees look at ancestry. They're saying here's the primordial ancestor, and from that primordial ancestor, here are the points of divergence to those organisms that are currently alive today. So it shows a common ancestor, shows development, whereas an unrooted tree doesn't show any of that development. It just shows current relatedness. So these are two different ways of looking at a phylogenetic tree, or two different styles of phylogenetic trees. And in both cases, the distance between the currently existing organisms, so here we would say A, C, B, D on both trees, are those that are currently alive today. And the length of the branches typically either shows the number of changes over time, or just the distance in terms of relatedness or relationship. So the internal node is showing an organism that no longer is alive today, whereas the external node is one that is alive today. And again, the branch is, the length may depict time or molecular changes, molecular differences. So lots of phylogenetic trees and plenty of practice to work through your understanding of those trees. And I suggest most certainly getting your hands on this article because really showing this to someone special in your life is going to blow their mind. So once again, just on the citation of this article, it's pretty easy to find because it was a pretty big deal when it came out. But this is in Nature's Microbiology, and A New View of the Tree of Life is the name. So in fact, I'd love to give extra credit on your final exam if you just write a quick note about something you learned when you went through and read that article. If you just put that at the bottom of the page dedicated to your answer, the Tell Me a Story answer. Marvelous. Let's take a moment to talk about this Bergeys Manual of Systematic Bacteriology. There are two editions, and the first edition is one that I have an old copy of this from the 1950s. I love having this on my bookshelf. You can see that it most certainly looks dated, but you can also tell that it's tiny compared to what we know now. So like within the 5 volumes of the new edition, there would be 3 of each this size or more. So we knew far less about the diversity of the microbial world when the first edition was written. Additionally, as you might guess, the first edition was almost entirely based on phonetic data, phenotypic characteristics, and there was basically one devoted to gram-negatives of medical and industrial relevance, and then one devoted to other gram-negatives, and then one devoted to gram-positives, and then another to all gram-positives except for actinomycetes, and the other one was the actinomycetes. So it was quite simplistic in terms of being all phenotypic, whereas now the new Bergeys Manual, the new edition, is all phylogenetics, so all based upon genotypic data. So there are 5 volumes in that new edition, and the first volume is actually sometimes fun-lovingly called the old ones or the ancient ones. And of course, you might guess that that's where we find that relatively little group of archaea. So the archaea are situated in Bergeys Manual Volume 1. And the archaea, of course, includes some of our favorite characters that live in places like Yellowstone. So sometimes they're thermophilic or hyperthermophilic. Some of them live down in hydrothermal vents. And a lot of them are sulfur metabolizers, though not all of them. We will meet in our Yellowstone virtual tour the Sophilobus genus, which is, of course, in the Sophiloballus order, but Thermoprotealis, Desulfurylcocalis. So the Euryarchaeota, another group, a much larger, a more encompassing group than the Crenarchaeota. This includes the methanogens and also the halophiles. So if you've ever seen a saltern, where salt is precipitated out of water in order to actually make salt, right, to produce salt for consumerism, you'll see that it turns bright red, and that's due to halophilic growth of Halobacterium salinarium, for example. So methanogens, the ones that grow in manure piles and make them bust into spontaneous flames, that's also in this group. So this is a very cool group as well. The deeply branching and phototrophic bacteria fit into volume 1 also. And they include some really auspicious characters. Thermatogae is a group that has what you might consider an outer sheath or toga, right, around them. They tend to be thermophilic as well. Deinococcus thermus, which one of my favorite types of bacteria, fits into this group. The genus and species Deinococcus radiodurans is extremely famous because Deinococcus radiodurans is capable of handling 3 to 5 million rads of radiation. Like, seriously, let me say that again. 3 to 5 million rads. Like, get this, the A-bomb, 1,200, right? Humans are very highly negatively impacted by 100. And these guys are taken 3 to 5 million. It busts apart their genome, scatters it into pieces, and they patchwork it back together. Think people are interested in Deinococcus radiodurans, aka Conan the bacterium? Oh yes, they are very interested in it. Chloroflexi, this is the next one. This bacterium has gliding motility. It's observed in Yellowstone. So in our mats, in our microbial mats in Yellowstone, we're going to meet this one. Aquaphysae, just like it sounds, Aquafex is a genus within this group that is water. It's a water maker, Aquafex, right? Makes water. Well, it uses hydrogen as an energy source, and in doing that, it creates water. Chlorobi, this is the green sulfur bacteria growing up in our Winogradsky column even as we speak. In fact, I was showing Cassie a layer of the green sulfur bacteria in her Winogradsky column, or in one of our Winogradsky columns, just yesterday. Cyanobacteria, also growing in the Winogradsky columns. Remember, these are oxygenic phototrophs. They grow at the top of the column where they extract electrons off of water and make oxygen gas. Let's look at volume 2. When volume 2 first came out, I got a notification in the mail saying, hey, you know, professor, buy this for your bargain deal, $396. So volume 2 contains all of our gram-negative unknowns, the proteobacteria, but also many others that fit into this volume 2. The alpha proteobacteria includes some that we talked a little bit about and some that you all have thought about, Brucella abortus, the very auspicious character in Wyoming. We've talked a little bit about Nitrobacter in context of the nitrogen cycle, and then Rhizobium you also talked about in lab regarding the nitrogen cycle, and it grows in symbiosis with legumes. Beta proteobacteria, we have mentioned Thiobacillus ferrooxidans. Remember my hometown of Leadville where it eats fool's gold for dinner? The gamma proteobacteria, by far the one we're the most familiar with because, hey, check it out. The families that we have worked with in lab, Enterobacteriaceae, where most of our gram-negative unknowns live, except for Pseudomonas, which lives in the family Pseudomonadaceae. Whoa. So amongst this whole vast 5 volumes of Bergeys Manual, and all of our gram-negative unknowns are in one class in one volume. That gives you just a little sense of the incredible biodiversity. Delta proteobacteria includes the predaceous Delovibrio, the bacterium that rams into other gram-negatives, bores holes in them, and eats them alive. And then finally, the epsilon proteobacteria, which includes the character we've talked a lot about, Campylobacter jejuni, that was the causative agent of my sister's watery diarrhea. Now, in volume 3, this is our gram-positives with low GC content. And we're looking at the Clostridia, Clostridium botulinum we've talked a lot about, in Clostridium tetani, and there are some questions about that on your final exam. The Mollicutus, which includes the mycoplasmas, as you guessed and know, these do not have cell wall, so they don't have any peptidoglycans, so you could think about what antibiotic you wouldn't use on them. Class 3, the bacilli, we're super familiar with these. This includes things like our lactococcus and our lactobacillus that we use in the production of things like yogurt. It also includes things like streptococcus that some of you had as an unknown, and we know some of which are pathogenic. Now this is weird. You're like, wait, why did she skip from volume 3 to volume 5? Well, when they were prepping to put these to press, what became volume 5 was supposed to be volume 4, and it kind of makes sense because gram-positives with high GC content comes next. So these would be the ones with higher melting temperatures, right? The actinobacteria, but this ended up being published later, so it ended up being volume 5, even though it would maybe make more sense for it to be volume 4. So just a thing. But this does include the actinomyces, which is a very large group producing a lot of antibiotics, right? Remember streptomycin, erythromycin, produced by streptomyces. And then lastly, in volume 4, we have some that vary quite a bit. They don't fit one category or another. So the planktomycetes are really unusual in their shape and also in the fact that they lack peptidoglycan. The chlamydiae, remember these are causative agents of certain STIs. And spirochetes, we've talked a lot about Treponema pallidum, also causes the STI, and has a corkscrew. Remember, the way that it gets through viscous solution is by moving like a corkscrew. And also bacteroidetes, which many of you are very interested in because they're very prevalent in the rumen of cattle. And so they play a really large role in that digestion there. So now that we are familiarized with these volumes of Bergeys Manual, we are going to take a virtual tour through Yellowstone, where we are going to meet the archaea and particularly focus in on Crenarchaeota and Euryarchaeota.